The most important part of working in a lab is having your safety gear, which includes a lab jacket, safety glasses, and of course gloves to protect your hands. Lots of times in the field, as the PE is out in the field, the PE can get very dirty, especially in the water. So to avoid contamination inside of the gas chromatographer and mass spectrometer, it's important to wipe down the PEs to get as much residue off as possible. After completely submerging the polyethylene sheet in its solvent, it's important to next add an internal standard. An internal standard is a mixture of deuterated and labeled compounds that are representative of the analytes of interest over relevant molecular weights. Adding an internal standard gives us an idea of how well the extraction goes after concentration. After the internal spike is added, it's important to cover the lid of the amber vial with tin foil to avoid as much evaporation loss as possible while extracting, and to seal it tight with a lid. Afterwards, the PE is ready to begin extracting and must be placed in either a box or some kind of drawer where sunlight cannot affect it, because the sunlight can hinder the extraction process. The solvent is then transferred into a turbovap tube, which is used to, a turbovap is used to concentrate the solvent down to a smaller volume. Now that your solvent is inside of the turbovap tube, it's ready to be concentrated down to about 0.8 milliliters. This is a turbovap where a nitrogen line comes from our tank inside of the turbovap, where it is blown over the solvent, and there's a water bath that evaporates, that is heated, which evaporates the solvent off, which concentrates the solution. By setting the turbovap to about 30 degrees Celsius and having the nitrogen flow over your samples, they're concentrated down. By carefully monitoring the volume of your samples, when the samples are about 0.8 milliliters, a little bit below 1 milliliter, they're ready to be taken out of the turbovap. Since we can't concentrate the samples any further inside the turbovap tubes, what we do is we transfer the sample into a smaller 2 milliliter amber vial, which can be concentrated further down with the mini-vap. Again, in order to ensure quantitative transfer, the 2 milliliter vial where the sample is concentrated by mini-vapping is drenched 3 times with solvent and transferred into the 2 milliliter vial with the glass insert. After transferring quantitatively into the glass insert, the sample can be capped and is ready for analysis by GCMS.